We've learned a lot about DNA at this point, what it is, how to replicate it, and how to make proteins using DNA as a code. So this chapter is going to explore DNA technology, and this is how we use our understanding of DNA to apply it to everyday problems. DNA is often collected as a very small sample that scientists actually need a lot of. So typically, we'll use DNA for any purpose. We need to make copies of it. One way to do this is by making what's called recombinant DNA. Recombinant refers to recombining. So this is combining different DNA of two different organisms. So typically a gene of interest, for example, the gene that codes for the protein insulin. So insulin is needed for diabetics, and so scientists make lots of copies of the gene for insulin. So the gene is inserted into a small piece of bacteria DNA called a plasmid. Right, so the plasmid is this circular DNA here, and the gene is put into that circle. Once the gene is inserted into the bacterial cell, then the bacteria are allowed to multiply. So here's a bacteria that's got a plasmid with a gene in it, and when that bacteria reproduces, it can make hundreds and hundreds of copies of that gene in hours. Then the gene can be removed. So we can either take the genes out themselves, or we can collect the proteins that are made as a result. So for the case of insulin, the protein insulin is produced by the bacteria and then collected to be made into pharmaceuticals. And then that insulin can be sold to diabetics. So how do you get this gene actually into the bacteria? So we have special enzymes called restriction enzymes. And those are the ones that actually cut the DNA. And they cut it through both strands, but they don't cut it evenly. So if you can see in this picture on the bottom left, it's got these little edges that stick out. And when it's cut with the same pair of scissors, meaning when you cut both with the same restriction enzyme, those edges will match up. And then when you insert the gene, it knows exactly where to insert into the DNA. So there are different ways of actually finding or making DNA samples to begin with. A nucleic acid probe is a piece of single-stranded DNA that has been labeled with radioactive isotopes. This works like a tag. It can be added to a DNA sample, and it will complementary base pair with the gene that the researcher is trying to find. Then the radioactive isotopes will fluoresce or glow, and the researcher can collect all of the samples that contain the gene that they want to study. Sometimes all that's available is RNA and not DNA. So we will use an RNA sample, such as some RNA viruses like the coronavirus. To make a piece of DNA from the RNA, researchers do a process that's the exact opposite of transcription. So this is called reverse transcription. So if you remember in transcription, we went from DNA to RNA. So reverse transcriptase is an enzyme that can go from RNA to DNA. And this creates a strand of DNA that's called complementary DNA, or cDNA, because it's made from RNA. One of the most powerful tools in DNA technology toolbox is the ability to edit specific DNA sequences inside of a living cell. CRISPR is a protein that allows researchers to cut DNA at a specific spot. This protein complex can be given a guide. This guide is an RNA strand that complementary base pairs with the gene of interest. So in my picture over here, here's my protein, and then this pink part is the RNA strand. And you can see it pairing with this part of the gene. This is what the DNA researchers want to cut. So this can be used to do what's called a knockout, which can break a gene, or it can be used to fix a gene. Scientists often don't know the function of a specific DNA sequence. So one way to figure it out is to break it and see what happens. So think in your house if you've ever tripped like a circuit breaker. So what do? You go to the circuit breaker and you flip all of the switches until you find the one that fixes the problem. So this is kind of similar to knocking out a gene. If a scientist wants to study a specific cellular function, let's say making a certain protein, What they can do is take all of the genes that they think are involved and knock them out one by one until they find the one that actually causes the change in function. CRISPR also allows for fixing genes, so once they've knocked out a gene, they can actually fix it. So if a gene was mutated or it was causing a disease, it would be super great if we could go in and actually cure it. This was done in mice in 2015, and it was actually able to correct a genetic mutation. But there are ethical barriers to be considered in attempting this with humans. Should we be editing human DNA? And it's never been shown in humans. The only research right now is currently with mice.